Of you. My name is Chris Dallin, and I am the marketing director for ASEP Inc. We have a very exciting company here, and we want to get the word out to as many people as possible so that you can potentially benefit from our success. A few housekeeping items. We use the Q&A function for any questions you may have. So please use that and we'll get to your questions as they come along. Just so you all know, the webinar is being recorded, and I will send you all a link to the recording within 24 hours, and you're very welcome to share that with your networks. We're happy to answer any and all questions, time permitted. We are scheduled for 1 hour. But we can go over time if necessary. The format is a little introduction from me, and then we were going to go on and have both Rudy and Bob introduce themselves. We're going to go through our corporate investor package or investor presentation today. There are some terms and some scientific vocabulary, which I think a lot of you would benefit from to get a better understanding of the company. And the one thing I wanted to say is that beyond our innovative and groundbreaking science in both advanced therapeutics and diagnostics, there's one really amazing thing about this company, and that's the people, an extraordinary team. And we have two very special guests today. Dr. Bob Hancock, who is the big man on campus at UBC. And on the other end of the continent, we have Mr. Rudy Mazzocchi, who is our CEO and big man on campus in Manhattan. That being said, I would like to pass the mic over to Bob and Rudy. Rudy's going to introduce himself, and then we'll move on to Bob. And so let's get started.
Great. Thank you, Chris. And it's a pleasure to be here. And thanks again for coordinating all this. It's done a fabulous job this morning. So I'll take some time. I'll leave a little bit of time left for Bob. I want to introduce myself and my background. Academically, I was actually a pre-med student at the University of Pittsburgh, where I majored in biochemistry. Actually, I was accepted at a couple East Coast medical schools, but opted to experience the California dream. I went to UCLA for graduate school there, where I had graduate studies in biophysics. And then I was recruited very early on in my career by corporate America into getting into the medical technology space. So I'm actually a co-founder of 17 companies, medical device, biotech, medtech, and recently biopharma. Holder of also co-authored over 110 patents because of my involvement in these numerous companies. And a majority of those resulted in M&A transactions. We sold actually multiple companies to the likes of Medtronic, Abbott, Johnson & Johnson, C.R. Bard back in the day, which is now Becton Dickinson, and several IPOs. I've actually taken companies from creation through the IPO process, both in the US on NASDAQ, the Australian exchange, and most recently now the Canadian exchange. And actually took several years off as a venture capitalist. I was a partner at a venture capital firm in Atlanta, Georgia called Acutive Medical Ventures. We also established something known as the Innovation Factory. We actually created and established another dozen companies from scratch there to take them through the clinical validation phase. And that's sort of been my standing mantra in these companies as I work with clinicians and researchers, the likes of Dr. Hancock, to bring these companies to fruition. I'm always seeking that early clinical validation, get through the regulatory process, make sure we raise the proper capital, build the team, expand the team, bring on the right board of directors, auditors, corporate counsel, and prepare it for that IPO or transaction. And as a quick example, we had a neurosurgical company called Image Guided Neurologics. We developed a technology to really enable the implantation of electrodes in the brain to treat Parkinson's disease. This is neurostimulation. We developed this technology platform, and early on we introduced the opportunity to Medtronic, who was the leading company in developing these pacemakers of the brain, if you will. And the process evolved to the point where, you know, I had a track record also of taking these companies to the commercial markets, and they ensured me that I was doing almost too much. What they wanted to do was control that process, control and be engaged in the early clinical development, the regulatory process, so they can define the endpoints, define how the product was labeled. So we negotiated a transaction, which is becoming more of a gold standard for what I do, is bring those corporate strategic partners in. We define milestones to achieve, they fund those milestones, and then at the end of the day, there was an acquisition. We actually teed up, we had a pre-agree on a valuation for the acquisition. So this is a model that I'm not saying we're going to try to replicate here, but we are speaking now to corporate strategic partners very early on. My belief is sooner than later. And we'll come back to that point on how that's generating revenue, how it's going to help us finance the company with even, you know, additional non-dilutive capital. So, and final thing is, along this way, because of that transaction with Medtronic, which we closed back in 2005, I was given the Entrepreneur of the Year Award by Ernst & Young. So I've actually been a multiple recipient of the Entrepreneur of the Year Award. And joining here, and I'm going to turn it over to Bob in a minute, it checks all my personal boxes, right? This is exciting, novel, you know, proprietary technology, a really rare consolidation of both diagnostics and therapeutics, which I seldom have seen in my career, working with a stellar development team and huge markets that haven't been addressed properly. So I'm anxious to be here, very excited to have you possibly join us and get on board with us. With that, I'll turn it over to Bob, and I'll jump in a little bit later as well. Bob is muted. And Bob, you're still on mute.
Yep.
There you go.
I'm not going to give a big introduction because you can read whatever you like on our website. But I am going to just briefly mention that I'm a fairly legitimate scientist. I'm in the top 250 most cited scientists in the world of all time. And I've won the major prize in antibiotic drug development. So I think I'm fairly legitimate scientifically, but I've also always been interested in the products of my research and where they can go. And so I've also been involved in company establishment, and I'm up to 5 companies so far, nowhere near Rudy's record, but at least I'm very interested in the other side of the fence. So what I'm going to do is just try to walk you through an enhanced version of the deck that we have on our webpage, because I'm excited about data and actual results that show clinical potential. And so I'm going to try and show you some of the results that indicate that we are not an early stage biotech company. We're significantly advanced. So I'm going to now share my screen. And you can see the theme of the company here, which is that we're fighting an issue called antibiotic failure. And I'll describe how this is a little bit different from pretty well what any other company is doing in the world. And we're fighting it on two fronts, both the diagnostic side and also the therapeutic side. And I'll describe these issues too. So as Rudy mentioned, we're a rare combination of both diagnostic and therapeutic technologies. And we're able to do this because of a couple of companies that I started as virtual companies called SEPSET and ABT that handle these 2 different areas. We basically existed for a long time, essentially on grant funding, a lot of grant funding. And I will describe that to you. But essentially, we have like $20 million of development money that's gone into this company already. So our purpose is to mitigate the global crisis of antibiotic failure and improve patients' odds of survival and the quality of life. So this gives you just some sort of like top level statistics. So the diagnostic area, which you'll always see in this kind of yellowish color here, is particularly directed towards a syndrome called sepsis. You may not be aware of how important sepsis is, although I'm sure it's touched almost everybody's lives. There are 49 million cases per year. When we're talking about early diagnosis, we can multiply this by 2 to 3-fold in the number of suspected sepsis patients. And this gives rise to 11 million deaths. So this is actually a staggering figure. You may not realize this because there are a lot of billions of people on this planet, but 11 million deaths is essentially 20% of all deaths on the planet in any given year. And in fact, it turned out to be more than this in the last couple of years, because it's now recognized that 100% of all of the people who die from COVID actually die from sepsis. So antibiotics are also, or treatment of bacterial infections is also a big deal. Basically, it turns out that the 11 million deaths, probably 1 million of these can be caused directly due to antibiotic resistance. And this is formal resistance due to the genetics of the bacterium involved. But there's another type of resistance that is really insidious. And this is biofilm-mediated resistance. And bacteria that form these lumps of bacteria on surfaces, I'll show you one in a minute, are about 10 to 1,000-fold more resistant to essentially every antibiotic. And yet, surprisingly, amazingly, there are no drugs approved for biofilms. And this is despite the fact that the FDA has recognized that 65%, nearly 2/3 of all infections are, in fact, biofilm infections. So most of the infections, but resistant to current therapies, are no drugs available. The markets are huge. These are the global antibiotic markets and the global sepsis diagnostic market. And you can see areas of this are in the $1 billion range, increasing at a quite significant rate. So I'd like to talk about the diagnostic technology first. The diagnostic technology involves a rapid diagnostic test using the same methods that are available currently in hospitals, a type of assay format called polymerase chain reaction, or PCR. And this is able to deliver results within 1 hour of a patient arriving in a hospital. And this will improve sepsis survival rates and will allow doctors to make better and informed treatment decisions. If we look at what happens, which is in this gray set of boxes at the bottom, we see that essentially a patient will experience illness and initial infection requiring hospital care. So they'll typically go into the emergency room either by themselves or in an ambulance. And the clinician, based on the symptoms that they see in the patient, which at this stage are really quite non-specific, will suspect there's the possibility of sepsis, and so we'll collect blood. And then the sort of gold standard test is to find bacteria in the blood. But in fact, the test results are not actually really definitive until 24 to 72 hours after that initial suspicion of sepsis. And the reason this is important is that for every hour you delay an appropriate diagnosis, there's a 7.6% increased probability of death. So when these conventional antibiotic therapies fail because of a delayed diagnosis, we see multiple organ dysfunction, and this results in death in 22 to 30% of patients, a staggering death rate. So it's almost like a death sentence. In contrast, what we see, what Sepset is offering is the same experience up to ER admission, but now when the blood is collected, within an hour, we can apply the Sepset early diagnostic test, and that tells the clinician whether that patient will go on to severe sepsis and whether that patient needs to be prioritized for immediate and appropriate treatment. So instead of delayed treatment, we have immediate treatment within an hour of entering into the emergency room. So these are some of the advantages and also some of the data that we're facing at this particular time. So we're in the final throes of developing the diagnostic assay so that we can go through a clinical study, and that allows us to file a 510K that then allows us to get approval. And this should all be possible this year. So this is data that you see at the bottom from emergency room patients, 313 of them all from all around the world. And you can see the performance of the test is really fantastic. So accuracy, 92%, sensitivity, which is the number of true positives, how accurately it recognizes whether patients have sepsis, 88%, and specificity, how accurately it diagnoses that a patient doesn't have sepsis, which means we don't have to start using our potent antibiotics for these patients, 94%. So this is way greater sensitivity and specificity than the criteria used today, which are these SIR symptoms, and also a thing called a quick SOFA score, which is a quick assessment of organ failure. And these actually have about a 50% accuracy. In other words, it's like flipping a coin. Because we get earlier recognition of sepsis at a time when patients are admitted to the emergency room, the physician can then now start to determine the most appropriate treatment really early on, and that decreases the risk of death. And we also have another next generation diagnostic coming along that will focus on the patient-specific immune response and will tell the physician exactly what type of sepsis that patient has. So we also have a potent antibiotic therapeutic technology. It's a proprietary peptide technology that directly addresses the ineffectiveness of current treatment options by suppressing biofilm growth and reducing inflammation. And I told you there are no drugs that actually work on biofilms. They usually use combinations of antibiotics, but these also don't work on reducing inflammation like our drug. So this is what a biofilm looks like, that green thing in the middle. And this is essentially a group of bacteria that grow on the surface in a kind of a colonial form. And it's a stress adaptation to growth on surfaces, and it can be surfaces in the body. And so they become resistant to essentially everything you can throw at this, including the host immune response. So what you can see over on the right-hand side here is the model that we built. It's called a skin organoid model. So we build a human skin in a dish. So you can see the filter, this gray thing underlying this skin. And the skin has all the regular layers that human skin does. And now we apply bacteria to the surface, which you can see on the left. And you can see these clusters of bacteria, which are this bacterium, MRSA, all over the surface of the skin. And I think you can see that the surface of the skin and the underlying layers are considerably disrupted. So it causes a lot of pathology. When we treat with peptide, which is what's shown on the right, it looks essentially like control skins. And you can see that these clusters of bacteria have gone away. So it works really, really effectively. So this addresses a huge unmet medical need. As I mentioned, there are no approved treatments for biofilms. There are more than 2.8 million antibiotic-resistant infections in the USA annually. And according to the mantra that 65% of infections are biofilms, 2/3 of these occur in biofilms that also become resistant due to the way they grow. I told you there are 11 million deaths globally from sepsis, and a lot of these initiate as biofilm infections. And it turns out that there are now these organisms called the escape pathogens. Escape is just the first letter of the bacteria's name that include untreatable variants. So one of these, methicillin-resistant Staph aureus, which is the S in escape, contributes to more deaths in the US per year than homicide, Parkinson's disease, emphysema, and HIV/AIDS. So it's really, really important. And I should say that the result I was showing you previously was MRSA. So these are some of the highlights before I show you just a little bit of data in a minute. We see potent activity against all major clinically relevant bacteria growing as antibiotic-resistant biofilms, and that includes the most resistant forms of these clinically relevant bacteria. We see strong synergy with antibiotics, so they work in combination with conventional antibiotics. They're safe and effective in animal infection models, including in vivo models of sinusitis and abscesses, which is a kind of a skin biofilm model. We see strong anti-inflammatory activity that is as good as the non-steroidal anti-inflammatory drug indomethacin, but a unique mechanism of action. We see also that while we're suppressing harmful inflammation, we also are able to boost protective immunity. And we have compounds that actually combine all of these activities into a single molecule. So our initial clinical opportunity in this space is going to be chronic rhinosinusitis. This is a lifelong condition causing painful inflammation due to bacterial biofilm infection. It's amazingly common. There are 242,000 emergency room visits in the USA alone annually. Antibiotics do not work, despite the fact that more antibiotics are applied to sinusitis than any other medical condition, because these antibiotics do not really target the biofilm, and they only treat symptoms, but not the cause. There's also a treatment now that has been introduced called dupilumab. It's a monoclonal antibody treatment, and it's the first treatment for inadequately controlled chronic rhinosinusitis. But there's poor evidence of efficacy, and it's massively expensive, which precludes the use in this chronic condition. So it's $30,000 a year, but it gives you an idea of the sort of market in which we're operating. Our peptides work as anti-biofilm agents and anti-inflammatories in animal models of sinusitis. And I just thought I'd show you a little bit of data here. So this is a mouse hooked up to a respiration device, and the mouse is knocked out. That's why it's kind of sitting in one place here. And what we've done is we've introduced into the nares of the mice, the nose of the mouse, bacteria that are engineered so that they produce luminescence. So you can see this huge light production here is due to the sinus infection. And when we treat with peptide after 5 days, we can completely make that light go away. So the bacteria are all killed. So you can see this actually in a graph. So here's the number of bacteria in the middle graph. We can see the number of bacteria recovered when we lavage the nares of the mouse. But when we treat with peptide, we see way less bacteria present. And the same time, we can see that this the inflammatory response due to this pro-inflammatory cytokine IL-6 is also substantially suppressed. Now, this is the first therapy ever to have both this antibacterial response and an anti-inflammatory response. We also have a second line that we're also actively in development with, together with our partner in the US, iFiber, and with funding from the US Research Material Command. And this is wound infections. So wound infections occur in war zones, and as you can imagine in the field, they occur in extremely dirty and unhygienic conditions, and they often become contaminated by bacteria that grow into biofilms, and these biofilms prevent healing. And so currently the only way to deal with these is to try to scrape out the wound, which of course doesn't help very much in healing either. So in collaboration with iFiber, a company in Ithaca, New York, and funded by the US Department of Defense, we've made wound dressings that can kill biofilms made by the most important wound pathogens. We've demonstrated these work really well in 2 species of animals. And we believe that this therapy that now is currently going to move towards clinical development also has huge potential in surgical dressings. And the wound healing market is $20 billion a year market. So this is not a market to be sneezed at all. We're not a two-trick pony, though. We're a multiple-trick pony. We have a lot of different therapies that we're developing. So this gives you all of the different sort of things. We've got 3 diagnostic technologies. It turns out that our sepset ER diagnostic assay that we're developing for severe sepsis in the ER, like I mentioned, is also great at diagnosing COVID severity. So we'll pick the individuals who end up in hospital that are most likely to cause the greatest problems to end up on respirators and to actually die. And also I mentioned this, we have this second diagnostic assay developed looking at immune deficits that can be used to guide therapy. And then we just have a whole range of different therapeutic options. I mentioned the top two, but we have a number of others that work in a variety of diseases, from inflammation through to veterinary, immunology, oral health, ID prevention, and so on and so forth. So we're extremely rich company that has all of these strategies. And Rudy already mentioned this, but the way that we're going to be able to develop all these strategies, despite the fact we're a modestly sized company, is through collaborations. And so Rudy's the guy with all the contacts. He's helping us to establish these collaborations. We've got a couple going already. The reason we're where we are is because we started from a really solid base. So this is all of this sort of non-dilutive grant funding that went into the research that makes up the two companies that underlie these two technologies. These companies are now majority owned by ASEP. So they're conglomerated, if you like, within ASEP. So a massive amount of money and this massive amount of money shows no sign of actually decreasing. So we're going to see this kind of investment continue on and provide, if you like, additional value to shareholders, because it doesn't dilute the value of the company. We have strong intellectual property, both in the diagnostic and the therapeutic area. We have a number of patent families, and we also have several awarded patents in both the diagnostics and the therapeutic area. So we're well sourced in that particular instance. And we also have a strategy, especially in the therapeutic area, to try to utilize orphan drug designation in order to speed movement through the clinic. That will mean that we can move through the clinic instead of in something like 8 to 10 years, we can go through in about 4 to 5 years. There's huge advantages to orphan drug designation. This is the share structure of the company. We're quite, at the current day share price, we're actually quite under capitalized, I would say. Most companies in our particular stage are in the hundreds of millions of dollars capitalization. And we anticipate that we'll see substantial growth once we get through the main things that are dragging back the stock market at this stage. So I'm going to leave it at that, but I'll just get Rudy to address this last slide to sort of summarize everything I've told you.
Great. Thank you, Bob. Yeah, and we'll address a couple of things I want to address. There was that money shot too, that slide that showed of our platform, as Bob indicated. There are several indications there that are multi-billion-dollar opportunities as well. I spent my early part of my career in catheter development for both acute and chronic catheters in patients, and the number one clinical problem was infection and sepsis. So excited to see that platform being developed. And to touch on as well, so this slide here shows that, these are enormous size markets. We have great patent protection already. We're going to continue to develop that intellectual property portfolio. There are opportunities with generating near-term revenues. As you've seen, we're going to be submitting a regulatory application to the US for approval of our diagnostics. We're anticipating we can, as early as the end of the year, have approval on that first diagnostic tool, which will generate potential revenues with a partner. But on the peptide therapeutic side as well, these corporate strategic partners, as we engage them, we're engaging them really to take this technology to the global markets, right? We have an obligation as a small team with limited resources to really take this to the millions of people around the world that can benefit from it. So the corporate strategic partnerships are extremely important, extremely valuable. And again, as I mentioned earlier, they like to get involved sooner than later so that they can drive the clinical endpoints, how the product is labeled, what the use instructions look like, even how they name the product, how they price the product. In exchange for that early involvement, we're going to seek milestone payments, if you will, followed by a royalty on the sale of the product on a global basis. But those milestone payments can be booked as revenue. They're going to contribute to our clinical development, our regulatory activities, and they'll be booked as revenue. So we're anticipating some early revenues here within the next 12 to 24 months. And we have sufficient capital today. If you envision both those technology companies that Bob mentioned, one on the diagnostic, the other on the therapeutic, they are independently, sort of view them as independent development teams that are well funded. We have capital in funding those development activities for the next 12 to 18 months. The parent company, ASEP Medical, has a runway of at least 12 months as well. So obviously we'll be raising some additional capital, but we have the time, we have the runway. In the public listing, we're also, as you know, obviously we're on a Canadian exchange. We're probably a couple of weeks away of being listed on the OTC markets here in the US. And we've qualified, we've passed for DTC eligibility. And we got to check a couple more boxes here and we'll be listed on the OTC market. The ticker symbol may be different. I think it's SEPSF. I'm working on trying to get that to be sort of correlate with our ASEP ticker symbol in Canada. I think that'll open up to a number of other both retail and institutional investors here in the US. And I have experience, limited experience, because I have more experience on taking companies, to an initial public offering. But I have the wherewithal and the contacts and the support and getting this to possibly uplisting on the NASDAQ. That's one of our objectives as well. And again, we couldn't do this without this spectacular team. It's a lean, small team. And again, I've worked with some of the best and most brilliant researchers and developers of technology in my past, but this is an exceptional team, which will continue to supplement and, we're bringing on other advisors and consultants, but we have the horsepower to get to where we need to go with the corporate strategic partnerships. So with that, maybe Chris, we want to open up for some questions.
Absolutely. We've got one from Nicholas already. He asks, what are the capital markets plan for the company over the next year or so? Is existing cash sufficient to see through the year for the company and to execute on the plan? Good question. And when do we expect these pipelines to be commercialized?
Well, I think I touched on many of those already. I think, the capital, the runway we have on the development side is quite sufficient for the next 12 to 18 months. Timing wise for the capital markets, as I mentioned here, with the OTC market, by the end of March. I think I'd like to beat that timeline, but it'll be at the end of March. And, we have engaged multiple firms now to support both the PR and IR activities of the company. We have a plan for news releases, press releases about every 2 to 3 weeks, as you'll see more and more coming here. Several key ones in this coming year will include activities surrounding our clinical activities, our submissions for, our regulatory submission to the FDA, and hopefully and potentially identification of 1 or 2 of those strategic partners.
Good.
Yeah, I'd just like to also touch on those questions. Firstly, we do have enough money to do the science part of the job. So part of the investment that allowed ASEP to become the majority owner of the 2 companies basically is sufficient for us to have about a 18-month runway. That should get us all the way through to approval for the diagnostic and definitely into phase 1 clinical trials into maybe IND filing and approval for the therapeutic. So those are essentially our commercialization plans. We plan to put a diagnostic technology on the market at the latest between the end of this year and early next year. And we plan to file an IND sometime in the middle of next year for our first line product, and likely the second line product about the same time, which is the wound healing.
Good. Have a question from Rachel. Are we considering a listing in Europe or Germany?
We haven't rolled that out. I have probably less experience there as well, but our focus has been primarily to get to the OTC and then, consideration for the uplisting to the NASDAQ. So Rachel, yes, we'll probably get back to you on that, but that's a feasible process in front of us as well.
Good. Next question is from Siobhan. Can you explain the process to get the FDA 510K approval for the Sepset ER? Is ASEP preparing to get approval with Health Canada or CE marketing in the EU? When is the typical time frame for these applications? When do you anticipate being able to commercialize Sepset ER diagnostic tool?
Yeah, these are all really good questions. So the 510K approval process is quite different from what we're all used to with therapeutics and normal for sort of phase 1, 2, 3 clinical trials. So essentially, it's a single approval process, and essentially it's a device approval. So what we do is we utilize existing devices, and we have a partnership with Thermo Fisher to allow us to develop our technology on their device, which is implanted in essentially every hospital in the US and Europe. And that we develop data for particular groups of patients, and that's all we have to do to get 510K approval. So currently, a lot of this we're doing through contract research organizations. So we have an organization that's advising us on the 510K filing process. That includes actually doing the 510K filing. We anticipate we'll have the data to go for that sometime around the middle of the year to maybe the third quarter. We also are engaging a contract research organization to help us coordinate trials in the USA. In most cases, the FDA is required to give you a response to your 510K filing within 4 months. And they can ask, of course, for additional data. That's where the uncertainties lie, but they can also give approval within 4 months. That also has happened previously. And your best chance of getting approval is to have your documentation really organized well. And that's why we hired a CRO to make sure that our organization of documentation is superb. So we are planning to get approval in a whole bunch of other jurisdictions, certainly with Health Canada and in Europe. But essentially the process is pretty well the same. And you can use a lot of the same data to do those filings. So we will probably, for those approvals, hire, again, expert contract research organizations to smooth us through the process of filing in these other countries. But we decided to pick the US because it's number one, a huge market. Number two, has the cash to pay for a diagnostic that in a lot of patients, which would help us a lot. Number three, has a huge COVID-19 issue still and is likely to have one for quite a while. And that's one of the major targets of our diagnostic technology. And I guess other countries, if you've got approval in the US, that smooths things over a little bit and you're getting your approval in other countries. I think I gave you the timelines there, basically.
Thank you for that question, Siobhan. Rachel has a question. I read the partnership news released with iFiber and funding support from the US Army. Do you have any other strong partners that you can name? supporting the advancement of either technologies?
Yeah, I can mention that we've had a reasonably long-standing partnership with Vetiquinol. It's the now I believe the 8th largest agricultural biopharma company in the world, and they are developing our molecules as immune modulators for treatment of companion and farm animal infections. So that's been going on for a while. We're also in discussions with a number of companies. So we're particularly interested in the oral health care area because we have superb data showing efficacy for oral care infections. And we're starting discussions with just the whole range of other partners. One of the nice things about the technology is because we have so many peptides available that this enables us to, if you like, license out different peptides to different partners so that we can do a lot of deals, not just a single deal. The area of diagnostics also, we're very seriously, so we've got, I told you about the partnership with Thermo Fisher, but we're also trying to engage a large partner because of the size of the area and markets and the also complexity of the diagnostics market. And so we're currently talking with the largest diagnostics companies in the world in a relatively advanced stage of discussions, but I can't make any announcements at this stage.
If I can just add on to Chris and Bob, a lot of the grants that you've seen identified in that one slide, there's continuation and follow-on grant opportunities. So they're not, although some come to completion, we're going to pursue, continuation funding from like the Defense Department, for example. There's other opportunities to expand that our reach for those grants. And keep in mind, they're all non-dilutive. It's a win-win for all of us when we bring in non-dilutive financing for the company.
Yeah, I should also mention that strategically for a company, it's better to take the technology as far as you can along the development process before you partner, just because the royalty income that you can make is larger if the product is better developed.
Good. Thank you, Rachel, for that question. Kenneth has a question. If we're looking at an OTC listing sometime before the end of March, do we have any idea about potential timelines for the NASDAQ listing? That's probably for Rudy.
Yeah, so the OTC status would be probably OTC QB. And we're looking at, maybe possibly upgrading QX as sort of a enhanced version of that. Process normally takes about 4 to 6 months following our OTC listing to qualify for the NASDAQ. And again, we probably have 9 out of 10 of the boxes already checked, but that is just working through the process. We'll probably look at a larger market maker here in the US to help us, to assist us with that uplisting.
Great. Thanks, Kenneth. Rachel has another question. Can the market get a rundown of the share structure, insider holdings, warrants, and options? We did have a slide there. Is there anything else we wanted to comment on that?
That's a good question. Part of the process with the OTC is that, we have to identify what we call our NOBO and OBO list, right? The list of shareholders. So that's underway. Part of the DCT eligibility requires us to complete that. So that's underway as we speak, being assisted by our CFO in that process. So that's in process, the best I can say right now.
I just comment on the insider issue because we all have to file insider trading reports, obviously. And I would say that everybody who is in serious management, and we also have a employee stock option plan, has a stake in the company, which I think is actually healthy, but nobody has a very large stake.
Thanks again, Rachel. Rachel has another question. You briefly touched on this, but do you have any large financial backers or in the future, have you planning any road shows to attract more brokers, funds, family offices, those kind of things?
Yeah, so real quick, we do have some large private individual holders. I don't know if we can maybe identify, I don't know if you recall, Bob, who the institutional investors might be, or hedge funds. But yes, we do have plans for sort of road shows. A lot of them will be virtual, obviously, today. We're doing additional webinars, podcasts, and again, there's going to be a stream of news releases up and coming. I'm also coordinating presentations at many of the larger conferences here in the US, including we're looking into one of the largest biotech conferences here coming up as well in California. So we'll continue to have a strong presence at those conferences. And, but we'll have to probably get back to you on that list of shareholders as well.
Yeah, I think the important thing is that we have a lot of blue chip shareholders, actually. We have a lot of people who are very highly placed in the Canadian banking and investment industry, and also in from other countries also. In terms of road shows, we're not going to do road shows at this stage until the stock market starts to recover well, because it's just not a good thing to be raising money when everything is so depressed. So, and because we do have a runway, it's not like we wouldn't, if you want to give us $10 million, we're more than happy to accept it. But what we don't want to do is end up diluting out the company for all the existing shareholders. So yeah, we're going to wait until the market starts to recover.
Good stuff. Thank you, Rachel. Siobhan has another question. What catalysts over the remainder of 2022 should shareholders be aware of?
Yeah, maybe I can just start this, but Rudy probably knows the answer a lot better than me. But obviously the catalysts are things that progress us towards obtaining revenue, early term revenue. And I'm talking about money that doesn't come in a dilutive fashion. So the progress of the diagnostic, I would say, is a catalyst. Filing the 510K would be a catalyst. Partnership opportunities would be a catalyst. And we're likely to have a fairly substantial, large grant funding opportunity to announce in the not too distant future too. So those are the sorts of things, is essentially things that feed money into the company but don't cost the shareholders.
Yeah, I think a lot of those are sequential too. For example, a large strategic partner might wait until we have verification of receipt of our application to the FDA. So we'll do those sequentially in parallel as we build those relationships with those strategic partners.
Good, thanks, Siobhan. Nicholas has a question, and I think this is a good one. NASDAQ has a requirement of $4 a share price for an uplisting. So how does the company plan to fulfill that requirement?
Two simple answers there. One is to do it organically, right? Is to build our share price, to enhance our share price based on announcements of our achievements and success, and grow it organically. The other way is that, if push comes to shove, you can always do somewhat of a reverse split. It's not an ideal situation, but many companies do that to get to the required share price. Our preference is to build it organically, and we have time to do that. As Bob indicated, we have the runway to do it.
Good stuff. Thank you, Nicholas. And Nicholas says thank you to you, Rudy.
You're welcome.
Question from Kenneth. And this question is for you, Rudy. Given your previous record of taking companies public and or brokering buyout deals, is a buyout opportunity to a larger company one possible exit strategy the team is exploring, or is the team fully committed to seeing the company through the development process? Good question.
Yeah, great question. And they go hand in hand, all right? We never like to build companies to sell them, but that's what usually happens. As I indicated, 8 out of 10 of my activities end up in M&A because they do want to, the large companies, the global companies want to control the technology, control the pricing, Again, we're going to do what's in the best interest of all of us as shareholders collectively. If a great offer comes along, obviously there's going to be a shareholder approval process to approve that transaction. I want to see us build significant value before that happens. There's ways of generating revenues. We can sustain ourselves. We have enormous a platform to be developed with several different indications in multi-billion-dollar markets. There may be a scenario where we have a strategic partner buy exclusive rights to a specific market, like the catheter sepsis market or the veterinarian market or for dental. So we have multiple bites at the apple here as well. But our key is just, we have time. It takes, as everyone knows, it takes time to develop a biotech company like this, to bring it to the commercial markets. We have the capital structure in place. We have the interest from the market. And now we just got to keep our nose to the grindstone and do what we need to achieve to build that value.
Yeah, and I think we could have also answered that by saying both are possibilities. In fact, both are even possibilities simultaneously. So in my opinion, you don't make yourself a candidate for a buyout by sitting in one place and saying, buy me. Obviously, you do it by demonstrating success in the development process. And because we have more than one string to our bow, it's quite possible that we could sell part of the company without selling the other part of the company, that we could still exist as a company, like say a diagnostic company or a therapeutic company, and sell the other part of the business. So I think we definitely have lots of potential here, but that potential will only be realized if we are successful in the development process. So that's our main strategy at this stage. It's just making sure that we're successful.
Yeah. Good question, Kenneth. Thank you so much. I'm looking here and it looks like we've answered all the questions. I would like to personally thank each and every person who attended today. We had over 40 attendees. And thank you to Bob and Rudy for your time today. As I said before, I will be sending along a link to the actual video so that you're welcome to share it with other people, friends and family. We will be doing these webinars on a regular basis in the future. So we think that this is kind of an electronic roadshow, and you can get updates from us on our progress as we develop the company and our therapeutic and diagnostic technologies. That being said, again, this company is about science, but it's also about great people. And I thank you all for attending today, and look forward to seeing you again.
And Chris, if I can add, if we didn't completely answer the questions proposed today, stay tuned next time. We'll have answers for you then.
Stay tuned. Perfect. Thanks again, everybody.
Thank you. Bye-bye.
Thank you. Bye-bye.